Okay, let's get started. So we're very excited about today's session because we're addressing one of the biggest bottlenecks in modern clinical trials, the black box of imaging operations. As trials become more global and protocols become more complex, managing imaging with physical shipments and relying solely on spreadsheets and emails is simply breaking down. The aim of today's webinar is to see how we can fix that. I'm Kire, the Senior Director of Customer Success here at QMENTA, and I will be the moderator of today's webinar. I'm thrilled to be joined by a panel of two experts who live and breathe neuroimaging. First, we have with us Dr. Daniel Ontaneda, MD, PhD, and full professor of neurology at Cleveland Clinic. Dr. Ontaneda brings a wealth of experience in neuroimaging trials. He is the co-PI of the flagship CAPS-MS study, which, as some of you may know, has enabled a paradigm shift in the diagnosis of MS using MRI. We're really excited to hear more from Dr. Ontaneda in today's webinar. And we also have Oscar Peña Nogales, PhD, our senior applied neuroimaging engineer. Oscar works directly on the front lines, helping imaging professionals, sponsors, and sites translate complex protocols into operational reality. Okay. If you've managed a neuroimaging trial across multiple locations, you know that as you add more sites, you invite more variability. We call this protocol chaos. It is the single biggest risk to your data and results quality. So how do we move from that chaos to scalable quality? Today, we are going to walk through an end-to-end framework for centralized, auditable quality control. We'll break this down into 4 key areas. First, we have the harmonization. We look at some best practices for standardizing protocols before the first patient is even scanned. Second is the verification step, where we'll show you how to move away from manual checks and use automation to catch errors instantaneously. Third, we have the oversight, where we discuss the architecture needed to monitor this at scale with full audit trails. And finally, we have the proof. We are honored to have Dr. Daniel Ontaneda share insights from the CAPS-MS study, showing these principles in actions. Our goal by the end of this session is to show you exactly how to scale multi-site imaging trials, cut operational costs, and improve quality across every single site. Before we dive into the webinar, a few housekeeping notes. You're all currently in listen-only mode to ensure audio quality. However, we want this to be as interactive as possible. So please type your questions in the question box in your control panel at any time during the presentation. We have a reserved time at the end for Q&A. Yes, this session is being recorded, and we will send the link of the recording and the slides to all registrants within the next 24 hours. Okay, so with that, let's get into the substance of the discussion. First, I will start with Dr. Ontaneda. Dr. Ontaneda, could you please tell us your experience with multi-site neuroimaging studies? What are the pain points that you faced, especially like in a flagship study like the CAPS-MS? Can you guide us through how that looks like?
Yeah, thanks so much, Kira. And I'm really honored to be here and share a little bit about our experience with multi-center imaging trials, and specifically the pivotal role that QMENTA played with making us make the collaboration across all these sites work. So we're in the business of clinical trials and imaging biomarkers, and as many of you who do multi-center clinical trials will know, is that harmonization across the sites is really of key importance. And you highlighted this a little bit, but the reality is that when we do clinical trials and we do biomarker studies, really there's a premium on making sure that the data that you're acquiring, which costs a lot of money to acquire, is done so in a way that is reproducible, where you can have trackable QC and QA that is done in real time, and that you can actually respond to difficulties at different sites. We work with premier academic medical sites, many of them, but the reality is that many times scanner upgrades happen, software changes occur, and you have to be able to respond to that. So that's kind of as a preface to say that, the imaging CRO side of what we do, I can't understate how important that is. And so, we embarked on this journey with QMENTA many years ago. We started out actually with a pilot study, and what we wanted to do was to demonstrate that the central vein sign can be used a useful biomarker for multiple sclerosis. And when we started out, we had, I think, 6 sites, and then we expanded out to 10, and then we expanded out to 11. And in the pilot, what we did is we actually worked with QMENTA to have basically a cloud-based solution for how sites would upload the images would undergo QC, the images would be pre-processed, finally processed, and then sent back to the adjudicator so that they could rate the central vein. And we did this successfully in a cross-sectional study that had a very small budget, but QMENTA were great partners in understanding that this was kind of the first step that we needed to take to make the central vein sign a reality. And so once we had that pilot data, once we demonstrated this relationship with QMENTA, and I have to mention that, the beautiful thing about working with QMENTA was the flexibility they offered for the types of solutions that we needed. So, some of the stuff, you come in, you know what you want, and it's pretty much what any imaging study needs. But then, at individual studies, for example, we needed specifically processing merging of the flare and the T2 star images. That's something that wasn't familiar, for example, to anybody. We created something with QMENTA that allowed our site PIs to rate the central vein sign. Once we developed some preliminary data from that, we actually applied for this funding through a U01 NINDS, which is the National Institutes for Neurological Disorders and Stroke biomarker pathway. And this was, I think, one of the first times that we were kind of validating a true neuroimaging biomarker for the diagnosis of a condition. And the stated goal of this application was to get the central vein sign into the diagnostic criteria. And this figure here that you're seeing shows kind of the complexity associated with running a study like this. The reality is that, it's not easy, but I think the fact that we had QMENTA as our imaging clinical data manager made things much easier. And QMENTA offered us a solution for MRI data and also for clinical data. And that was something key. When we did the pilot study, we had only focused on MRI data, mainly because our budget was a little bit low. And I can't tell you, that was a huge headache for us to manage that in a red cap. It was layers of complexity, and getting queries sent out to the sites was really complicated. So we learned our lesson in the pilot. And so for the actual U01 study, the clinical data also flowed through this QMENTA algorithm. And as you can see here, these were the sites that did the analysis. Once QMENTA had the data, we had to feedback the data back to the site so that they could do the CVS rating, which was all done. And basically, to us, it seemed very simple. It was a shiny, glossy solution to a problem that we had learned took us a lot of time. And then, of course, the results get dumped, and that went to our central statistical analysis. And the actual results of the MRI data went to Cedars-Sinai and NINDS. And we basically are now in our last year of the study. And to be honest, I think the main kind of take-home message is that we got the central vein sign in to the diagnostic criteria, which was our ultimate goal, and I don't think we could have done that without QMENTA.
Thank you, Dr. Ontaneda, very much for your valuable insights. I'm sure your opinion will resonate very much with what Oscar will be presenting next, as he will guide us through this end-to-end framework, capturing the steps that basically were applied in real time, in real life, in the CAVZN study. I will now hand over the mic to Oscar. Oscar, the floor is yours.
Yeah, thank you very much, Kire, for the warm introduction. and also Dr. Ontaneda for the insightful topics you covered. Okay, so now let's look at what we face on the ground. So we often think of clinical trials as these clean scientific experiments, but operationally, they are chaotic. You have competing clinical priorities at hospitals, a very high turnover of technologists, and among other things, unscheduled vendor upgrades and that change the scanner software and hardware halfway through the study. So if we ignore this operational friction, we end up with unknown data failures. These are, for example, protocol deviations, calibration gaps, and subjective quality assessments. These aren't just annoyances, they are a hidden debt. If we don't pay attention to them early, it is very likely that your study will end up paying for the consequences later on with unusable data. And this cost is actually measurable. For example, we know that up to 50% of the imaging time points in a clinical trial contain some type of error. So we see attrition rates are climbing, and that between site variability in multi-site studies reduces the study efficiency by up to 12% compared with single-site studies. So now let's look at the literature. So whether it's pediatric gliomas or Alzheimer's disease studies like ADNI, the conclusion in all of them is very, very similar. So some examples. So the top left one, the addition of multimodal imaging modifies the assessment in a small number of cases. Top right, only 1/3 of MRI requests meet recommended acquisition criteria. Bottom, if you rely on different acquisition conditions, your variance increases. So as you expect, all these discrepancies will harm the statistical power of your study. As such, we have to move from hoping for quality to engineering the quality into the study via every possible way. So the critical question is, when does QC start? So most people think QC only happens after the scan is performed. That is, let's call it the reactive approach. When anyone has 10 spare minutes to assess its quality, the patient has already gone home and the money is spent. We need a preventive approach. QC starts the moment you write the first draft of the imaging protocol. It starts by considering the reality of the sites, not issuing a generic PDF, but defining tolerances, balancing speed with quality, and standardizing the hardware before a single patient is recruited. Remember, documents are static, but site reality is dynamic. Hence, our job is to bridge that gap between documents and sites in order to guarantee the successful execution of the study. So this brings us to the life cycle of quality. As you can see on the left-hand side, the end-to-end image quality life cycle is not a single step. It is a continuous loop throughout the whole duration of the study. Quality begins with site selection, choosing sites based on capability, not just patient volume. Then it continues with site qualification by calibrating the scanner before the first patient walks in. Then the golden subject, with the purpose of validating both the acquisition setup and the subsequent workflow on a human subject. Once the trial starts, we move to continuous monitoring and the execution of both manual and automatic quality workflows. And if at some point an issue with a site or a patient is identified, the loop might have to start all over again. So I am actually very happy to say that this loop is what the QMENTA personnel and platform facilitates. So now let's break down the foundation of this loop and explain QMENTA's role in it. Okay, so let's start with the foundation. Best practices to harmonize imaging protocols. Site selection should always start with a site qualification questionnaire, and its content must be carefully reviewed to guarantee the required minimum expertise and capabilities of the site. I have created this very poor example by asking the AI, and as you can see, the questionnaire is very, very incorrect. So now in this slide, I saw a large number of aspects that must be taken into consideration during the setup phase of the study, particularly in relation to protocol design, site selection, and site qualification. I'm not going to go into the do's and don'ts as it will take us the full hour, but let me highlight 3 of them I consider very relevant. So first, guarantee patient conform. Second, use phantoms throughout the whole duration of the study. And third, do not allow a variety of hardware only to meet the targets. Now let me walk you through 2 real-life examples. So here is an email exchange. a technologist in Barcelona uploaded a phantom scan that had artifacts. Because we, the QMENTA platform, monitor this centrally, we caught it immediately. We didn't just reject the scan. We investigated further and realized that they were using a wrong phantom for the field of view defined in the study. Hence, we guided them to the correct equipment. Without this active loop, they might have scanned patients with the wrong setup. This second example shows a DTI sequence. As you can see, the site has set the facing coin direction to right to left. However, the protocol mandated anterior to posterior, as indicated in the sequence name on the top. This change completely alters the geometric distortions of the brain, blocking the execution of distortion correction methods. I have to say that we got this parameter deviation at source, hence preventing the error from affecting any patient. But once the study is ongoing, we cannot check every parameter from every acquisition manually. In order to scale, we need automation. So this is the QMENTA platform. Think of it as the operating system for your trial's imaging data. It handles the entire journey, from secure data upload to the identification, automatic quality control, central review, and finally, data analysis. The QMENTA platform includes all of this in a single compliant and cloud-based environment. As a result, it facilitates the management of the study from beginning to end in a very seamless way. So the process in the QMENTA platform starts with a smart uploader. This isn't just a file transfer tool. It is the key to guarantee scalability. When a site drags and drops a scan into the QMENTA platform, we run a battery of checks. We scan for viruses. We de-identify the data by removing all the PHI. We identify and harmonize the metadata in order to perform a deep learning-based classification of the imaging data uploaded. Overall, this will facilitate management of the study while maintaining compliance. And critically, last, we automatically execute the quality protocol adherence analysis. This analysis checks the DICOM headers against imaging protocols previously defined during the setup phase of the study. So the protocol quality adherence analysis looks like shown in this slide. So we can apply over 50 different rules instantly, applicable to the repetition time, the echo time, or the flip angle, among others. The protocol adherence analysis can also check data integrity and specific tags of each vendor. We can literally check everything. Next, as soon as a condition fails, for instance, in the example on the right, the voxel size, instead of being 1 by 1 by 1, is 1 by 1 by 1.2, the system flags the data session immediately and sends a notification to the user. So this approach avoids human error and saves weeks of time. Also, and very importantly, the site gets feedback immediate, not hours or days. But unfortunately, still in 2026, fully automation is not possible. Algorithms are perfect at checking numbers, but they still cannot replace the human eye and expertise. So this is why we have a visual QC step. In this visual QC step, the expert looks for motion artifacts, completeness of the field of view, spike artifacts, and incidental findings like unexpected tumor, among other things. We do this in the QMENTA platform, which is an integrated workspace. The viewer, the scoring form, and the query system are on the same screen. No extra spreadsheet, no need to download files. And very importantly, the QMENTA platform is compliant with all the needed regulations. Okay, so now let me show you what the QMENTA platform looks like in practice. So for the sake of the presentation, I previously recorded this very short video. I will be speaking over it. Okay, so I am now in the QMENTA platform where I have a project manager role. What you see here is the dashboard for a demo project, very similar to the CAPSEMS study Dr. Ontaneda mentioned earlier. Right away, I have a clear oversight of everything. I can see the projects I collaborate in and the total number of subjects, sessions, and analysis of this demo project. I can further go into the data tab. So here is our list of patients. You can see the status of every single time point, which ones are completed, processing, or failed. Hence, I don't need to email a CRO to ask, how is enrollment going? As I can see the real-time status of the study right here myself. For example, I'm going to open now one subject. I'm going to show the information of all the visits and then select the corresponding MRI form. So as you can see, on the right side, we have the MRI data uploaded. And at the bottom, we have the protocol adherence check. Remember, we talked about blocking errors earlier, right? So this is it. The system automatically checks the DICOM headers against our defined rules. Green check means it passed the tolerance ranges and everything matches the protocol. If this were red, a query to the site will be raised immediately. So now let's look at the analysis tab. So this shows the pipeline the data travels through. As you can see, there are many steps here. Some are automated algorithms and some are manual checkpoints. For today, I want to focus specifically on the image quality control step. This is where the human expert interacts with the data. So while the images load, notice the layout. On the right, we have the medical viewers showing 6 different images. And note, these images aren't just raw data. They have already undergone some automated preprocessing, which, by the way, is vital for identifying biomarkers like the central vein shine in MS. Then, on the other hand, on the left side, we have the digital case report forms. So, as you may be aware, this determines the efficiency of your trial, because many times a radiologist has a PACS workstation on one desk and an Excel sheet on another. So that constant switching between screens might cause error. So here is integrated. Of course, I can also zoom in a window, zoom out, and scroll through the sequences right alongside the question. So I'm going to quickly review the sequences. I'm checking for artifacts or incomplete coverage, but everything looks clean. So I'm going to mark everything as excellent. Then I have to click submit. Once I click submit, two things will happen. First, the pipeline automatically triggers the next step, which can be the generation of a report or some biomarker quantification. Second, every click is timestamped and logged in the database. So that means that 6 months from now, if an auditor asks who approved this scan and when, we have the exact digital record linked to the image. And of course, this is how we move from protocol chaos to a streamlined auditable workflow. So now let me move on to the following slide. So we have looked at a single subject, right? So now how do we manage a whole study? So to manage a whole clinical trial, we move from serial processing to global concurrency. You have to keep in mind that data management within the QMENTA platform is fully in the cloud and fully automated. This means that while some sites in some parts of the world acquire data and transfer it to the QMENTA platform, later on the same day, expert reviewers in other locations can perform the needed visual quality control checks and any needed reads. So this 24 hours non-stop active workflow means the study never sleeps, removing bottlenecks, reducing burden to sites, and most importantly, reducing cost. Also, very important, all the steps performed are traceable and auditable, as I mentioned before. So finally, given that the QMENTA platform is a centralized system, both project managers and PIs, we provide for them a real-time oversight, as we showed before in the quick demo video. You don't need to ask for a status report. You can log into the system and see it yourself. You can track enrollment, monitor site performance, and drill down into longitudinal data for specific subjects. For instance, you can see outliers instantly, which sites are failing or which subjects are drifting, among other things. Overall, this allows you to manage the trial proactively, fixing problems while they are small. And with this, let me give back the microphone to Kire. So, Kire.
Thank you, Oscar. Thank you for guiding us through this end-to-end framework. And I would like now to get back to Dr. Ontaneda. As you already talked about the CAVS-MS study and the complexity of it, I think it would be very interesting for the audience to hear an update on where the study is at the moment and see some numbers that represent this complexity. And even more importantly, if we do a what if scenario, what if this framework was not deployed for the CAVS-MS study? What is your opinion on the quality of the data and the possibility to deliver the results that, as you already mentioned, have been accomplished?
Yeah, absolutely. Well, thank you so much. And maybe what I can illustrate is how the processes that Oscar just highlighted, how they work kind of in a real world case of a study that used the QMENTA platform. And, this is kind of the study visit data entry, and just overviews the number of visits that we had. So we had a total of 420 participants in the study who were seen at 5 different time points. So baseline, month 6, month 12, month 18, and month 24. We're almost finished with our study now. We only have approximately 3 visits pending. And naturally, when you do these types of multi-center studies, especially with people that come for a diagnosis of MS, some of those individuals won't have multiple sclerosis and will disengage a little bit. So, of course, you'll have a little bit of patients who get lost to follow-up. But I just want to reflect that across these visits, there was a fair amount of queries that were generated. And I think when you generate queries, it means that you're probably doing your job right? We are identifying problems on the go. So a total of 750 queries were issued through the course of the CAVS MS study. And the majority of those, 55%, were for incomplete or missing data. And so we were able to respond to those in real time. It's very different if a coordinator, for example, puts in an incomplete or missing data point and you reach out to them a day later or 2 days or a week later versus if you wait a year, which is typically what happens when you're not using software that allows you to do queries in live time. And we'll go into a little bit of the results of what those queries look like. And then inconsistencies, if we can go back one on the slide. Inconsistencies and clarifications were approximately 40%. But interestingly, the number of queries that were related to MRI were actually quite low compared to the clinical queries and the other data queries, so only 5%. And I credit the very robust pipeline that QMENTA has for the very small number of MRI queries that were generated, and these were related to missing data, incorrect file names, and relaunch analysis. And we can go to the next slide. So a little bit into more detail specifically on the MRI, I think really what we wanted to highlight here was the fact that we completed a total of 420 MRIs at baseline, 318 MRIs at month 24, and the initial QA was actually passed in the vast majority of these cases, so 98.1. And you can see, both at baseline and at follow-up, once an MRI passes the QA, the likelihood that they're going to complete the pipeline flawlessly is really high. So 100% in the baseline data, 99.1% in the month 24 data. From then, we actually had the MRIs go to board-certified neuroradiologist who actually did an actual visual check of the quality of the data, specifically the T2 star imaging. And you can see once again that that was 100% at baseline and 90% at month 24. And once again, after queries passing neuroradiology QA actually goes up to 99.5% for the baseline visit and 98.3% on the month 24. So very clearly this was working. In terms of our neuroradiologist completing the CV rating, once again 99.5% at baseline. They haven't completed it all at month 24, and that's just up to a delay on our neuroradiologist. But when you look at the PIs, it was 100% at baseline, and at month 24, because the neurologist doing the CV rating don't do it unless the neuroradiologist is completed, they're always a little bit delayed. So we're doing that analysis right now. And then, of course, we did a central CV rating, which was completed in 100% of the patients. So you can see here, this is kind of what the month 24 QA looks like in a little more detail on the column on the right side. And, we had a proportion of 20% of individuals who were lost to follow-up, a couple people who refused MRI, refused gadolinium. But the total number of individuals who were failing QA with all these processes put in was only 1.6% of patients. And they failed because of scan quality in approximately 0.9% and a wrong protocol in only 2 patients. And so, when you think about doing an imaging study that requires you to, your primary endpoint is imaging as it was in this study, the fact that we were able to get failing QA in only 1.6% of all the study visits is really a testament to the way this system works. That is, the query is generated, it cannot pass through initial QA, it will not complete the pipeline. We get notified immediately. We go back to the sites, we ask them to rescan, we ask them to fix the protocol problem or the machine problem in real time. And really the results are here. Our data quality for this study are really superb. I've done imaging analysis now for over 20 years, and I have to say that this is the type of thing that I dream of when you start a study that data looks as clean as it does with this project. Next slide. Um, and I think this is kind of key learnings from the QMENTA side as well. But I would say that in terms of what we've learned is that forming a collaboration or a partner with a company that is flexible, that understands your needs, and that has built-in processes really led to the success of our project. I would say that our ability to be successful with this would have not been possible, to answer your question, Kira, had we not had this support. I don't think we could have reported back on our baselines. The data that we used was presented at the diagnostic criteria meeting in Barcelona, and this is what got the central vein sign into the diagnostic criteria in part, and I don't think that would have been possible without our collaboration with QMENTA.
Thank you so much, Dr. Ontaneda. It's really great satisfaction for us who have been involved on the front lines of making this study a success. It's really satisfying to hear that this has resonated so much with you and the rest of the users from the CAPSMS study. To wrap it up, today, just to go over, we explored the journey from protocol chaos to scalable quality. We saw how the QMENTA neuroimaging platform safeguards the entire study lifecycle, from the initial harmonization of protocols to the nuances of hybrid automated and manual QC. I want to extend a special thank you to Dr. Daniel Ontaneda for grounding these concepts in reality with his insights from the CAPSMS NIH-funded study. We hope you leave here with a clear roadmap. Everybody, the audience, leaves with a clear roadmap for achieving project oversight at scale. Okay, so I think that we have some time for answering a couple of the questions that have been shared by the audience during this session. Let me just give me 1 minute. Okay, so the first question, you mentioned before that QC should follow the preventative approach, which is partly focused on standardizing the protocol across all sites up front. However, can we not acquire data the way the site prefers and then fix the site variance later on the pipeline? So I believe this question refers to the statistical methods that kind of take into account possible variations and try to take these confounding factors into account to fix for the results. So, Oscar, I'll defer this question to you. What would you say?
Yeah, very, very good question, actually. So there are various statistical harmonization techniques available, both actually, to harmonize the acquired data as well as to harmonize the results, the final results. And while they are used in academic research, I personally advise against using them in the clinical trials for a couple of things. So first, they are not a magic wand for poor quality or inconsistent data. That's not the one said, yes. Second, statistical analysis might be able to correct for a certain degree of systematic and predictable bias, but it might not be able to correct for all subtle differences. Or if it does, we might not be able to guarantee the computed biomarkers are completely accurate, right? Third, sometimes it might not be possible to apply them just because of the corresponding study endpoints. And I think there is one more thing. So yeah, these approaches might change the statistical power of your study, which will result in needing to include more patients, resulting in an increased cost. So in short, I think personally, upfront standardization ensures the basic data is captured safely, and I would say it is a must, it is required. Other applications, while possible, I think they have to be done with a lot of care. Yes.
Dr. Ontaneda, what about you? And I'm sure that you've reviewed many papers and applications that talks about this approach. What is your stand on this? Doing the quality once the data is acquired and trying to fix for the errors afterwards?
Yeah, I mean, I think this comes up a lot when you do longitudinal imaging studies, and I agree completely with Oscar. That is,
Mm.
you want to try to optimize as much as you can, standardizing the acquisition of the images, and dealing with the lack of harmonization that occurs on the back end. If that involves a large proportion of your population, it really threatens the results of your study, and it threatens all the effort that you've done to try to do an adequate analysis. However, I will comment that doing longitudinal imaging studies, this will always happen to one degree or another, right? Um, you know, there was always going to be places where they are replacing scanners. It happens. There will always be software upgrades. They happen. And then there will always be human error. I think the key is having a system in place that allows you to track those in live time so that you can troubleshoot them on the fly, right? So even if you have a scanner, for example, that's changing, which you could say, I have no control over that. But figuring that out at the end is going to be hard versus if you figure that out immediately when it happens, you can go back to the site, ask them for scans that are done before and after, and apply a correction that might be impossible to do if you're doing at the very end of the study. So I think having the knowledge and the understanding of where things are in live time, I think that's super valuable.
Thank you. Thank you both. And we have one more short question, which is quite interesting considering how hot of a topic AI is. During the presentation, you mentioned that current AI approaches are not yet sufficient to fully automate the visual QC workflow. With all the recent advancement in AI, what actions are being taken in this area, and when can we expect the full unsupervised automation? So yeah, I would like to extend this question and to ask, do you think that we are anywhere near, or how long before we can remove the manual visual QC that has been an integral part even in the Kafzeme study?
I'll take the question first. So yeah, I think actually it's a very, very good question. So, and in my opinion, both industry and academic research are investing a massive amount of work into pursuing automatic QC. Actually, we ourselves, QMENTA, we are currently training advanced deep learning models specifically designed for motion identification, detection of anatomical cropping, and other artifacts and other issues that happens in both in CT and in MRI. However, in the short term, I do not expect a timeline for full unsupervised automation where AI makes the final QC call without human intervention. Because when dealing with regulatory grade clinical trials, the stakes are very high, right? So currently, the human eye and the clinical judgments remain irreplaceable for handling nuances of medical imaging. And answering your second part of the question, so I personally believe that in the near future, what we will see is a shift towards AI-assisted QC rather than AI-replaced QC. So in this case, these algorithms will act as a highly efficient first-pass filter. They will flag a specific slide telling the reviewer that they have detected some artifacts, but ultimately, human verification of the AI step will remain mandatory to ensure the integrity of the data. Yes.
Thank you.
Yeah, I mean, I
Dr.
would agree.
Ontaneda?
When we
Yeah.
did our UO1 study, as part of our grant application, we put in that there would be human review of the scans from neuroradiologist perspectives to ensure, data quality. I think what we've demonstrated is that, if you have a robust pipeline, the actual process of the QA actually is pretty high, right, in terms of the percentages of the scans that got approved by the neuroradiologist, and the QC goes smoothly. But I agree with Oscar that doing that in a fully automated way is perhaps aspirational at this point, and especially if you're dealing with federal funding or anything that requires regulatory approval. there is a requirement to have a set of eyes on things. So that step remains important.
Okay. And one more question came in, Dr. Ontaneda, for you. You mentioned 750 queries sent to sites. What is the main consequence of this number? Is it lost time, cost, concerns about reliability of the results?
Yeah, I mean, I would say that, the 750 queries are 750 problems that we avoided, right? And that includes missing data that would have not allowed a correct adjudication of the diagnosis of MS, missing data that would have not enabled us to complete the analysis as was set out in the grant application. And in my mind, those are opportunities. They do create work, if you like, right? You have a query, it gets generated, we flag it, we send that to the sites. The sites have to respond in a timely manner. But I would say that, if you don't do those, the cost is that you have data points that are missing. And if a key data point is missing, it's hard to know what data point is going to be key depending on what patient you have. Essentially, you're effectively reducing the power that you have to detect, a given difference in your study. So in the business of clinical trials and in the business of imaging biomarkers, that basically is lost money. So it's protecting your investment of your research idea if you ensure adequate response to the queries. I think that answers the question, Kira.
Yes, yes, yes. Thank you, Dr. Ontaneda. Thank you, everybody. Thank you, Oscar, as well. Thank you to all the attendees who stayed with us during this webinar. As I already mentioned, the recording and the presentation will be sent to you within 24 hours. So thank you, everyone, and have a great rest of your days. With this, we close this webinar.